Okay. Hello, everyone. I'm Yingying Zhang. I recently completed my PhD at Dr. Haiwan Yu's lab at Cornell University. And today I'm going to introduce our work focusing on deciphering the functional effects of somatic mutations in cancer by integrating the distinct perspectives provided by 3D protein structure and the topology of protein-protein action network. So our study specifically tackles two challenges in cancer biology. So first, among millions of somatic mutations in large cancer cohorts, how to identify those small set of functional ones which drive the development of cancer, we term them as driver mutations, from the vast majority of passenger mutations which are simply bystanders during tumor development but are not functional. And second, beyond identifying cancer driver mutations, it is also very important to understand their functional mechanism so that we can bridge the gap between the small mutations in the DNA and the malignant transformation of cells. So to tackle these two challenges at once, we need to combine the insights from different fields. And on one hand, this mutation rate modeling gives us statistical insights into which specific genomic regions exhibit significantly higher observed mutation rates than a background, so that this can help us identify driver mutations. But furthermore, to understand the functional mechanisms of the driver mutations, we really need to, can you hear me? Okay. So to further understand the functional mechanisms of the driver mutations, we really need to combine the insights from another two fields which provide complementary views. So on one hand, structural biology enables us to map the spatial organization of mutations on 3D protein structures so that we can better understand their molecular level effects. And on the other hand, so the systems biology provides us with a broader view of how those mutations disrupt the nodes and edges in the biological network so that they impact the systems level function. So in this study, we develop NetFlow 3D, which is a unified computational framework to identify and unravel the functional mechanism of driver mutations. And the first part of NetFlow 3D is to identify driver mutations. So this is achieved by integrating mutation rate modeling at genomic level and the 3D structural analysis at protein level. So this integration results in the identification of those significant spatial clusters of mutations on 3D protein structures, which suggest accumulation of driver mutations. Many existing studies have demonstrated that identifying spatial clusters of mutations either within single protein structures or on the binding interface between interacting proteins is very effective in identifying cancer driver mutations. However, existing algorithms have two major limitations. So the first limitation would be that they usually obtain an empirical background mutation distribution by randomly shuffling the observed mutations within the same protein structure. So this essentially assume a uniform mutation rate everywhere. So this is conflict with what we know about the highly varying local background mutation rate along the genome. And the second limitation is that these methods typically select one structure per protein or per protein complex. And so that only represents a single static snapshot of one protein. So we really need to account for the dynamic natures of protein configurations when we perform such type of clustering analysis. So our 3D clustering algorithm address both challenges, both limitations by first, modeling the highly varying local background mutation rates along the genome, accounting for covariates including replication timing, mRNA expression level, Hi-C, which describes 3D genome structure, as well as local gene density and local GC content. And this approach is inspired by Lawrence et al., which has been a gold standard in cancer genomics in modeling background mutation rate. And on the other hand, when we generate a contact map of mutated residues, we define the physical contact between every pair of amino acid residues based on the smallest 3D distance across any available configurations. So this approach can largely summarize different configurations of the same protein. And as a result, our 3D clustering algorithm achieves state-of-the-art performance in both identifying intraprotein 3D cluster as well as interprotein 3D cluster on the binding interfaces. And more importantly, so those 3D clusters demonstrate very high enrichment for catalytic residues, while those other mutated residues that are outside these 3D clusters show a significant depletion. Next, in the second part of NetFlow 3D, it further interprets the larger scale functional effects of those 3D clusters, which we identify in the first part. And it achieved this by further integrating the insights from the protein-protein interaction network so that it can identify the specific modules within the network where different 3D clusters are significantly interconnected with each other. And to give you a more detailed view of how this is achieved, so specifically we identify 3D clusters within every single protein as well as on the binding interface of every known protein-protein actions so that we can gain a global view of how these local 3D clusters within the protein structure are globally organized in the network so that we can identify their convergence on common modules in the network. So it is precisely because of the unique combination of 3D clustering analysis and the network topology so that we name our entire framework as NetFlow 3D. So the establishment of NetFlow 3D hinges upon the human protein structurome, which is a comprehensive repository that we compiled that contains the 3D structural data of every single protein as well as the binding interfaces of all known protein-protein interactions in humans. So the vast majority of these 3D structural data were generated by recent deep learning algorithms. So specifically, we leveraged the single protein structures generated by AlphaFold 2, as well as the partner-specific binding interfaces generated by our lab's deep learning framework called Pioneer. So it is because of the comprehensive nature of this so-called structural realm, which really enables a full coverage spatial mapping of mutations on 3D protein structures. So this allows us to identify any existing 3D clusters, both on experimentally resolved structures as well as on the vast majority of deep learning generated 3D structural data. So this is the key to obtain this global view of the network organization of these local 3D mutation clusters, which is the foundation of NetFlow 3D. So impressively, we found that more than half of the 3D clusters we found were identified using deep learning generated structures. And here I want to show you an example of a 3D cluster within a single protein structure generated by AlphaFold 2. And notably, none of the residues within this 3D cluster mutate in more than 2 tumor samples, meaning that these residues individually are less frequently mutated so that they can hardly be identified by recurrence-based methods. So the AlphaFold predicted structure is really the key for the identification of this 3D cluster. And here is another example of an interprotein 3D cluster on a binding interface between 2 interacting proteins. And this binding interface is uniquely generated by our Pioneer deep learning tool. So when we evaluate these 3D cluster generated using deep learning generated 3D structural data, we found that they are also highly enriched for catalytic residues, which are comparable to those 3D clusters identified using experimentally generated 3D structural data. So this gives us more confidence about the quality of our 3D clustering analysis using those deep learning generated 3D structural data. And after comprehensively identifying 3D clusters, then we try to identify the convergence on common network modules. And this is achieved using a heat diffusion model. So specifically, we assign initial heat scores to those proteins, to every protein, based on the significance of their 3D clusters. And then we iteratively diffuse the heat along the connecting protein-protein actions until it reaches a steady state. And then by compare the overall initial heat distribution with the steady state heat distribution, we can calculate the net heat transferred between proteins so that we can identify interconnected modules. So here I want to emphasize that an interconnected module is one where every pair of proteins, any pair of proteins have 3D clusters that significantly impact each other. In other words, we require within the same module this very dense and pairwise bidirectional impact. So this approach is to avoid the identification of star graphs, which center around very well-known cancer proteins have very significant 3D clusters, but including a lot of surrounding proteins with minimal 3D clusters and biological relevance. So since the NetFlow 3D integrates 3D clustering analysis and network topology. So first, I want to demonstrate the advantage of NetFlow 3D over simply performing 3D clustering analysis. So the advantage manifests in two aspects. So first, the dense interconnections among different 3D clusters within the same module reinforce their validity. So bolstering our confidence in the molecular level discoveries. So this is demonstrated by the fact that these significantly interconnected modules demonstrate much higher enrichment for non-cancer genes compared to those proteins that are identified solely based on 3D clustering analysis, although the latter has already demonstrated pretty good enrichment. And the second advantage is that by extending our analysis beyond molecular level analysis, we can get a broader insights into what are the underlying dysregulated biological processes. And this is demonstrated by our observation that these NetFlow 3D identified significantly interconnected modules have much higher consistency with established biological processes compared to those random group of proteins with 3D clusters. But importantly, this higher consistency is not simply an outcome of the network topology, because when we randomly select some connected components from the network, these connected components still demonstrate much worse consistency compared to the NetFlow 3D identified modules. And then I want to demonstrate the advantage of NetFlow 3D over those methods that relying solely on network topology. But before that, I want to highlight what are the key differences between NetFlow 3D and standard and traditional network analysis. So first, since the vast majority of mutations in cancer are actually passengers, so those 3D clusters really suggest the accumulation of driver mutations. So if we strictly focusing on 3D clusters, this can highlight cancer pathways which have been previously blurred by the overwhelming passengers. And in NetFlow 3D, the heat source are defined based on the significance of 3D clusters per protein, while in standard network analysis, a generally used way is to use gene mutation frequency as the heat score. And on the other hand, previous studies have shown that functional disease mutations on the binding interface are more likely to disrupt the corresponding interactions than a non-interface mutation. So inspired by this biological knowledge, when we model the impact of 3D clusters on a specific binding interface, we deliberately assign additional propagation weight when we perform the heat diffusion along the corresponding edges. So, but in standard network analysis, since they generally don't consider which mutation is on the binding interface with which interaction partner, so they will simply diffuse the impact of 3D mutation clusters to every neighbors equivalently. So as a result, NetFlow 3D achieved a much higher statistical power than standard network analysis. And to demonstrate this, I will systematically remove the 3D clustering information which were originally integrated in NetFlow 3D. So first, I removed the edge weight. So the edge weight in NetFlow 3D is determined by the significance of 3D cluster on the binding interfaces. So in this way, the resulting significantly interconnected modules contain only less than 1/3 of proteins compared to the original NetFlow 3D identified modules. And on top of that, I further removed the NetFlow 3D the initial heat scores that NetFlow 3D identified to each protein, which is just to replace them with gene mutation frequency. And in this way, the resulting modules contain only 1/8 of the protein compared to the original NetFlow 3D identified modules. So this result shows that by incorporating more and more 3D clustering information, which is provided by protein structure, we can really boost the statistical power. And then I evaluated the biological and clinical significance of NetFlow 3D identifications. And as you can see, our NetFlow 3D identified modules have similar levels of enrichment for non-cancer genes compared to well-known cancer pathways, while these enrichment levels are significantly higher than general biological processes curated by gene ontology. And furthermore, those patients with NetFlow3D identified mutations have significantly shorter survival time than patients with other mutations. And across all these cancer types, the hazard ratio are larger than 1.5. So in summary, NetFlow3D identify driver mutations in the genome and then map them to 3D clusters within protein structures, and these different 3D clusters converge on common network modules. So by applying NetFlow3D to TCGA pan-cancer data, we generated this multiscale functional map of somatic mutations, which cover a broad spectrum of biological processes and cellular functions, such as immune responses, signaling pathways, chromatin remodeling, etc. And with that, I would like to thank my advisor, Haiwan, and Dr. James Booth for their valuable input in this project, and all the co-authors, as well as the funding source. And I'm happy to take any questions.
Thank you very much. Are there any questions?
Very nice talk. Really enjoyed your talk. So I have a question about your mutation rate variation modeling.
Mhm.
You use like this mutation rate variation on the scale of 1 kb, but you are trying to predict actually drive mutations. So you really need this very good model on a single nucleotide resolution.
Right.
So what did you try to do that? This is the first question. I usually ask two questions.
Okay.
So in the second question, actually, how, when you evaluated your method of driver cancer prediction, mutation prediction, you showed survival curves, yeah?
Yeah.
So I wonder how other methods perform, or just say mutational frequency recurrence. Would it perform as well as your clustering? But I'm sure clustering will help. So really enjoyed your talk.
Oh, thank you, thank you. So I think both are very good questions. So for the first question, I think currently we can't really achieve those base pair resolution mutation rate modeling, because all those replication timing or mRNA expression level, those are none of them are at base pair resolution. So currently, what we are trying to do is, for example, in Mute6CV, they are like putting each gene have 5 features, like those 5 features, so that they put it in a feature space, and then for each gene they take the 5 a few bunch of nearest neighbors for to estimate the background mutation. Yeah.
There are this trinucleotide, pentanucleotide signatures, but I can talk to you.
Yeah.
There is a way to estimate the single nucleotide resolution background rate
for
cancer. Yes.
Okay.
So anyway.
Okay. Yeah. So sorry. What is the second question?
Yeah. The second question about your driver mutation evaluation. Basically, you evaluated survival rates.
Yeah.
Great. Yeah. But what about other methods which predict driver mutations?
Okay.
So you compare with them and say, mutational recurrency, what clinicians usually use.
Oh, okay. Yeah. So I think if you use hotspot identification methods, those simply recurrence-based methods, actually it can give you more striking difference in survival, actually. But our 3D clustering analysis is really to help to rescue those driver mutations that are like not frequently mutated, so that we can improve statistical power. They might have smaller effect size, but really we want to comprehensively identify all of the driver mutations, so that if we see like decent survival difference, then we think this is we're pretty satisfied with it.
All right.